Well, hello students. I hope you all are doing good and you all are in good health. What we are going to study today in this video lecture is post-fertilization changes. Okay. Now, post-fertilization changes, what are they and which part of your flower changes into what after the fertilization? Let's see. So, this is a typical flower that we are drawing here. Now, after the fertilization, what happens is the ovary, okay, changes into fruit, and the remaining floral structures moved on the either on the end of the fruit or remain connected to the thalamus. It depends on the position of the ovary, whether it is uh, superior or inferior. Okay. But apart from the ovary, remaining all structures, okay, in most of the cases are dried up. Okay, so remaining all structures dry up and the ovary swells and transforms into fruit. Now inside ovary, the ovule also transforms and they become seeds. Correct? So what we are saying here is after fertilization or post fertilization, the ovary is transformed into fruit. Okay, and the ovule is transformed into seed. Now let's see how this ovule or what the different structures of ovule are transformed into which part of the seed. Okay, so let's see, this is a typical ovule, and the ovule has this integument. The outer integument converts into outer seed coat, which is known as testa. The inner integument converts into inner seed coat, which is known as tegmen, whereas the primary endosperm nucleus is converted into endosperm. The zygote is converted into embryo. Now, endosperm and embryo both unitely forms seed kernel, whereas the outer and inner seed coat, that is testa and tegmen, fuses to form seed coat. Okay, and the seed coat and seed kernel both together forms seed. Okay, great. So now you can see how the different components of ovule are transformed after fertilization to form seed or to become seed, as well as how the ovary transformed into a fruit. Not only this, what you see here, the micropyle of the ovule remain intact and become the micropyle of seed. So in every seed, what you see is the presence of a microscopic pore. Actually, this pore is micropyle. Okay, and this micropyle in seed has a role, and this role is that it provides a passage for intake of oxygen and water. Correct? Very good. What else we find is if there is nucellus, okay, right? We have spoken earlier that in an ovule, the nucellus may be present or may not be, correct? So if there is nucellus still present in an ovule, after fertilization, the nucellus gets converted into perisperm, okay? And these perisperms are edible, they are nutritious, as we find in case of black pepper or in case of beet, the seed of beet. Okay, very good. Well, now let's see how these changes takes place once the fertilization occurs in an ovule, how these changes take place one by one. Okay, so what I'm drawing here is an ovule, is a fertilized ovule to be precise. So there is a there is a zygote. So egg cell has converted into zygote, which is diploid, and there is a PEN, primary endosperm nucleus, which is 3N. Correct? Now, immediately after fertilization, the double fertilization event, what happens is zygote undergo a resting phase. Okay, so the zygote does not immediately starts to divide and develop into embryo. No, it does not happen. The embryo, the zygote undergo a resting phase, and only after this resting phase, the development of zygote occurs to form embryo, not before that. Okay, so the zygote is undergoing a resting phase. However, the PEN or primary endosperm nucleus does not go in the resting phase, and it immediately starts to develop into endosperm by the process of mitosis. So the cells of the nucleus of PEN undergo mitotic division to form endosperm. Okay, great. So let's see what endosperm is. Right? So when we are talking about endosperm, let me tell you, endosperm is a nutritive material, or you can say it is the food store of the seed. Okay. Now, what plant does is it packs enough food into the seed so that it can, or the seed can nourish itself until it grows its own leaves and start to do photosynthesis. Okay, in other words, what you can, or what you can take analogy of, it is like uh, the endosperm is like the food source. Okay, when you are going outside, your mom packs your lunch box to uh, give you ample of food supply until you return back, and this uh, lunch box, or the food in the lunch box, is packed accordingly. Correct? So is the case of seeds. Inside the seed, plant packs enough food so that it is adequate for the seed to germinate. Okay, it is adequate to give energy to the growing zygote until it starts to do photosynthesis on its own. Okay, and in different plants it is different duration, and accordingly the seeds of that plant have more or less endosperm or food reserve. Okay, great. Now, since endosperm is nutritious, we use it for nutrition. So whenever we are eating seeds like gram, pulses, rice, wheat, whatever we are eating actually for their endosperm, because this endosperm packs a lot of food, a lot of nutrition. Okay, let's see. Now, endosperm is part of seed kernel. Okay, and seed kernel is only found in seeds. That is, it is only going to be found in plants which produce seed, or so-called seed plants. Now, you know that from the plant kingdom that seed plants are only gymnosperms and angiosperms. Okay, you have studied that gymnosperms produce naked seed while angiosperms do not. Correct? And only gymnosperm and angiosperms are spermatophyta, that is the plants which produce seeds. Okay. Now, so we find endosperm only in case of gymnosperms and angiosperms. Correct? Now, when we are talking about gymnosperms, in gymnosperm, double fertilization does not take place. Double fertilization is a characteristic feature of angiosperms, and that's why in gymnosperm, the endosperm is haploid, because it develops from unfertilized cells, or unfertilized cells of the ovule. Okay, याद रखिएगा इस बात को. It is very important. In gymnosperm, the endosperm is haploid, or haploid endosperm is found, while in angiosperm double fertilization takes place, especially triple fusion, as a result of which PEN is formed, which is triploid, and from PEN, endosperm is developed, and hence the endosperm is triploid. Okay, so do remember this, that in gymnosperm, endosperm is haploid, in angiosperms it is triploid. Okay, इससे बहुत बार क्वेश्चन पूछा गया है, and how they ask this question is not that they are going to directly ask कि angiosperm में endosperm की ploidy level क्या होगी, diploid, triploid, haploid क्या होगी. No, they will not ask you this straightforward. क्या करेंगे क्वेश्चन में? क्वेश्चन होगा कि suppose a plant or angiosperm plant has endosperm having 48 chromosomes. What is the number of chromosome in its pollen grain, right? Or it can ask questions like, suppose there is a plant whose pollen grain contains 16 chromosomes. What will be the number of chromosome in its endosperm, considering the plant is an angiosperm? Okay, I hope you all can solve it. If any of you have doubt in it, you can contact me directly. So we saw what is endosperm. Now let's see different types of endosperms, right? So when we talk about different types of endosperm, the first type is nuclear endosperm, right? Now what is nuclear endosperm? See, I'm taking this ovule, and in this ovule we said that the zygote undergoes a resting phase, whereas the PEN immediately starts to divide. Now, in case of nuclear endosperm, the PEN does not show complete division. Rather, the nucleus only undergoes nuclear division. There is no cytokinesis. As a result, this cell becomes multinucleated. Okay, so see, I'm making many nuclei, and all are triploid nuclei. So only nuclear division is taking place, correct? As a result, what is happening? This entire thing is one cell, and in this cytoplasm, so cytoplasm is a liquid, and this liquid has a lot of nuclei, multinucleated condition, and this entire space, this which is supposed to be endosperm, is a liquid in state, right? So this endosperm is liquid in state. Do remember, nuclear endosperm is liquid. Now PEN, free nuclear division, as a result multinucleated condition arise, and the multinucleated condition is also known as synocytic condition. Okay, now example is coconut milk. So once this nuclear division is done, the complete set of nuclear division happens. After that, cellularization starts. Okay, it is not that nuclear division followed by cytokinesis, then again nuclear division followed by cytokinesis. No, this happens in normal cell division. In case of nuclear endosperm, firstly the nuclear division occurs, the karyokinesis occurs, free nuclear division happens, and multiples, thousands and millions of nuclei are formed. As a result, this synocytic condition appears, as you find in case of coconut milk, and after that cellularization start, and then as a result of cellularization, cytokinesis happens, and formation of uninucleated cells of endosperm occurs. Okay, and that's why what you see is slowly the liquid endosperm is converted into solid endosperm. This is the process of cellularization. Okay, now this cellularization can happen either from the center or from the periphery, correct? So either this from in this entire liquid endosperm, this process of cellularization can start either from the center or from the periphery. Now, in about 60% of the plants, that is majority of the plants, the the cellularization of the nuclear endosperm starts from the periphery, okay, not from the center. Now, example is coconut or maybe cotton, wheat, maize, sunflower, etc. Okay, so this is nuclear endosperm. Now, the nuclear endosperm is the most common one. In most of the plants, what we find is nuclear endosperm. However, there is another type of endosperm that is called cellular endosperm. And as the name suggests, in cellular endosperm, the primary endosperm nucleus undergoes complete cell division. That is uh, complete mitotic division. That is the karyokinesis is followed by cytokinesis. Okay, it is not that karyokinesis will occur in a row and then cytokinesis will happen, that is cellularization will happen. No, it does not happen that way in cellular endosperm. In cellular endosperm, nuclear division, then again cytokinesis, that is cell division. So from one cell, two daughter cells will be formed, not only nucleus. As a result, the cellular endosperm is solid in state. Okay, so formation of uninucleated cells of endosperm or solid endosperm is formed. Okay, not very common. It is only found in about 72 dicot families, such as uh, Annona, Adoxa, you know, Adoxa, Datura, and Aristolochia, not Annona, Adoxa, Datura, and Aristolochia. Now, most of the plants which show cellular endosperms are saprophytic and parasitic in nature. Okay, so cellular endosperm. The most common one is nuclear endosperm, and then is cellular endosperm. Apart from this, there is another type of endosperm that is called halobial endosperm. Okay, so the halobial endosperm, it is actually the mixture of both nuclear and cellular endosperm. What happens here? The PEN undergoes partial nuclear and partial cellular division. Now, how it happens? Let's see. What happens is the PEN first divides to form two nuclei and followed by cytokinesis. This cytokinesis is horizontal. As a result, two chambers are formed, chalazial end, and that is chalazial chamber, and micropylar chamber. Now, mostly the chalazial chamber undergo cellular endosperm. So the development of chalazial side shows cellular type of endosperm, whereas the micropylar side shows nuclear endosperm. Okay, like development. So see, if you see the chalazial side, there is complete division. As a result, solid cellular endosperm is formed, whereas if you see the micropylar side, only free nuclear division occurs. As a result, the nuclear liquid state endosperm is formed. Okay, now mostly the liquid endosperm formation precedes the cellular endosperm formation in halobial. Now, so this is it. Now, where it is found, it is mainly found in monocots. For example, Erymuris. Okay, याद रखिएगा ये example of Erymuris. Apart from these three, there is another special type of endosperm that is known as ruminate endosperm. Okay, ruminate endosperm. Now, what is it? It is a special type in which was discovered by an Indian botanist called Periyaswamy. So Periyaswamy Indian botanist हैं और इन्होंने discover किया था ruminate endosperm. And what it is, how it looks, you will find that the innermost area in such an endosperm or in such a seed is uneven and multi-layered. Okay, where it is found? It is found in case of betel nut, जिसको आप हिंदी में सुपारी कहते हैं, और it is found in case of Palmaceae, the family. Okay, the family of palm. So date palm, है ना, fan palm, ये जितने भी palm के सीड्स होते हैं, उन सीड्स के अंदर जो एंडोस्पर्म होगा वो कैसा होगा? रूमिनेट टाइप, या फिर अनोना, एटसेट्रा। इन सबों में आपका जो एंडोस्पर्म होता है वो रूमिनेट टाइप का होता है। Now, just to understand, look at this picture. Can you see there is white part? The white part is endosperm, and this is uneven and multi-layer. This is ruminate endosperm. Okay, so this is all about endosperms. In next video lecture, we will be talking about the development of zygote. Okay, so stay tuned and stay healthy. Thank you.